nervous system. Hopefully you guys are excited. The nervous system is epic. I don't like it as much as I like the immune system. No offense to the nervous system. But if you didn't get to see my immune system screencast last week, definitely check it out. Get that 5 Plus membership. Watch the replay. Just trying to eat a cookie. Get signed up with that. Okay, but nervous system, it's got to be my second favorite if I had to rank all of our body systems. And hopefully by the end of this, you see. Okay, so purpose of the nervous system to transmit signals throughout the body and react to our environment, right? That's a big thing for us to be able to react and respond. Like when someone calls your name, you can process that signal and analyze it and respond. When you touch a hot stove, you can react and respond to that. When it gets cold, when it gets warm, all of these things, our nervous system really permeates, as does our immune system and our endocrine system, which is why they're the 3 systems on the AP exam. Those 3 systems really permeate every other system as well. So they're really important. So also, obviously the nervous system controls our body and communicates throughout our various body systems, is what that should say, not symptoms. Although if you have symptoms of something, your nervous system is also working. Okay. And we'll take a look at all things action potential, neurons, all that good stuff in the slides to come. Pretty cool. Okay, so we're going to start out pretty broad, and then we're going to get very specific when we talk about how an action potential propagates and you actually think, move, all that good stuff. So just from a broad standpoint, two major parts of the nervous system, central and then peripheral. And you've probably heard of these before. They're pretty straightforward. So the central nervous system is just our brain and spinal cord. All of those are going to be wrapped in cerebral fluid. We actually talked about meningitis a couple of weeks ago now, maybe a month ago. And it's called meninges, the fluid that's actually around our brain and spinal cord. So if that gets infected, you can obviously see why that would be such a large problem. So that's our central nervous system, and then aptly named, our peripheral nervous system is peripheral, or comes off of that. So you can see all of our other nerves on the right-hand side of your screen coming off of the brain and spinal cord. That's going to be our peripheral nervous system. So all of our sensory and motor neurons are going to come off of those major stems. Some neurons interact directly with the brain, but most are going to go through the spinal cord. But it really just depends on where in the body that they are. So like our sensory neurons up here in the eye, ear area, they're just going to interact directly with the brain. Something from the finger would go up through. I'm realizing that I'm tracking my mouse on the wrong slide. So they would interact, like eyes, ears, interact directly with the brain. If it was like your finger feeling something hot, that would go up through the spinal cord, into the brain, back down through the spinal cord, out, and then we'd signal a motor neuron to pull that hand away so that you don't get burned. Okay, so all of our nerves come off of this. Easy enough. All right, then there's the neuron itself. So the neuron is the cell of the nervous system. Looks kind of funky. You've probably seen it before. Okay, so we have dendrites on one end. Those are our signal receivers. So they are the ones who are receiving signals from all over. This is like your typical neuron, but I'm going to show you on the next slide. Like sensory neurons don't look exactly like this. Motor neurons don't look exactly like this. This is just like the most basic version of these things. Okay, so dendrites, you can see we can actually receive many, many, many signals. They're going to go through the cell body or the soma, where the nucleus is held, and then they're going to be propagated down the axon. Okay, so the axon is going to send our signal, and then we have axon terminals that are going to then deliver that signal to potentially a number of different places. But there's only really one pathway out of this. We have to travel down the axon. So we're going to see this shortly, but it's really an all or nothing response. Once the dendrites receive a signal and decide that they would like to respond, it's all or nothing. Okay. So axon terminals down here we'll talk about at the end. They're going to be the ones who are actually going to pass the signal on then to either a dendrite of another neuron or perhaps to your muscle cell or any number of body cells. So that's what those guys are at the end. Another really important thing are these wrappings. They're called the myelin sheaths. And if you know anyone with multiple sclerosis, that's actually a degeneration of the myelin sheath. So we can talk about that in a little bit. But basically, the myelin sheaths wrap around the axon, and our axon kind of acts like an electrical wire. So if you picture like a live wire, like a wire that is active and charged, if you touch it, you get shocked, right? But we wrap it in insulation like plastic, and then you touch it, you don't get shocked. So myelin sheaths are essentially that insulation. But you'll notice there's little gaps in the insulation, okay? And this actually allows us to propagate a signal much, much quicker. So signal can actually bounce between these little exposed areas of the axon, as opposed to having to run through the entirety of the axon. Hi, Addie. And because of that, our neurons can travel at 330 miles per hour as opposed to 11 miles per hour. So it makes a very big difference that we have the majority of our axon actually insulated, so that we only have to travel amongst these parts that are not insulated. Again, it makes us about 30 times faster. And you can run 11 miles an hour if you're trying. You certainly can't run 330 miles an hour. So it makes we wouldn't be alive if we didn't have our myelin sheaths. So if you know anyone with MS or have heard of it before, that degeneration can be really devastating, and eventually they will not survive just because their neurons are firing too slowly with the loss of that sheath. Any questions so far with the basic structure here? Ooh, uh-oh. Can anyone else not hear me? That would be terrible if I was just talking at you. I guess asking by saying is not a great move. Oh, okay. So other people can hear me. So Costa, I would just check maybe your sound, or maybe log out and log back in. Like my go-to solution for everything. Turn it on, turn it back off. Oh, an unmute button. Perhaps. Take a look around. Let me know if you still need help. So we're good so far with structure here. Again, pretty basic. A lot of you may already know all of this pretty darn well. Some of you may not, so I'm going through it anyway, but we'll amp it up real quickly here with talking about action potentials. Okay, cool. So directions of message is this way, dendrite to axon. It doesn't move in the opposite direction. It's unidirectional, and we'll see how that works in just a few. Okay, here's what I was talking about with the different types of neurons and what they look like. So you have your sensory neuron here. So sensory, relay, or interneurons, more frequently they're called that, and that's what they would be called on the AP exam, and then motor neurons. So motor neurons look pretty much identical to what we were just looking at. Motor neurons are what moves your muscles, right? They allow you to motor through. You can see here, this is a classic example of a muscle fiber. That's what our muscle fibers look like. They're very stringy, and they have those little banding patterns on them. And so they're receiving a signal and then transmitting it via these axon terminals to this muscle. That's motor neurons. They move your muscles. Relay, or interneurons, are basically going to, so they have a lot of dendrites and a lot of receptors. I kind of picture them, they're mainly in the brain and the spinal cord. And I basically picture those as like old-fashioned like phone operators, right? So the sensory neuron, calls in, and they're like, hey, we need this muscle to do that. And they're like, okay, let me connect your call, right? And they're like processing the information. And they're like, hey, motor neuron, sensory neuron needs you to move your hand. My oversimplified version of explaining an interneuron, right? But it comes in between, and it interpolates information, right? So it's taking information and then processing it and getting the right people the information that they need. So that's your interneurons. And then again, mainly brain and spinal cord. And then sensory are going to be the ones that actually sense things. So they have usually special receptor cells, which depends on what type of sensory neuron they are. If they're in the eye, or if they're in the nose, if they're in the ear, all really, really cool neurons that are interacting there that look a little bit different and act a little bit different. But they're going to have some sort of receptor at the end, as opposed to like your typical kind of dendrite axon business. So just kind of interesting, kind of cool. They look a little bit different. Okay. All right, so 2 basic pathways that you just need to be generally familiar with. So anytime we're processing information, so you hear a sound and then there's an outcome, right? Like your teacher calls your name and you turn your head, or you see something and you start running, right? There's a sensory input. It goes to be integrated into the brain, and then there's a motor input, okay? Motor output. So this is going to take a longer time than what we're going to see next, which is more of our reflex, right? And the main thing is that we're going from the peripheral to the central to the peripheral nervous system. So we're sensing, we're integrating, and we're motoring. Okay, this is just an important pathway to be familiar with, for sure. And nervous system comes up in a number of ways on the AP exam, but that's going to be in juxtaposition to our reflexes, where we're actually just staying essentially in the peripheral nervous system, or only touching central for a very short period of time. So in a reflex, oftentimes, like when you go to the doctors and they bang your knee and your knee just like involuntarily swings, right? Instead of going all the way up to the brain, we're actually just going to go from the sensory neuron. I don't know where this picture originated because they spell neuron weird. But the sensory neuron into the spinal cord, that's what this is. This is an actual, so cross-section of your spinal cord looking from above. So if we were looking down at your spinal cord and just with one little disc, that's what this would look like. And so it just goes up into your spinal cord and back to the muscle, as opposed to going up to the brain to be integrated, which is kind of interesting. And so that's why your reflexes are going to be so much faster, because they don't have to get integrated. There's no real analysis to be had. It's just kind of what happens when certain sensory neurons are stimulated. Okay, so basic pathways, but with all of these, there's going to be some sort of stimulus. There's going to be your sensory neuron integrated by usually an interneuron. It's going to be outputted by a motor neuron, and then there's a response. And again, this all happens in a fraction of a second because your neurons are flying around at 330 miles per hour. Okay, easy enough. Just basic pathways here so that we can see how these guys work together before we zoom in really, really, really small and talk about the membrane of the neuron and how we send an action potential. Okay, so we're finally there. You know the basics now. So we can talk about how we transmit actually a signal. So when I think about transmitting a signal, and this is a very common analogy, right? When our brain is trying to tell us to do something, or when our sensory neurons are picking up on something and a signal's being integrated, it's basically like pushing down a stack of dominoes. Okay, so if you think about that, right, there's a couple of reasons why this is very similar, and I'll make the connection for you as well. The start, right? When you're starting to push some dominoes over, right, you can lightly blow on the domino and it might be like wobble a little bit, right? But once it gets a strong enough signal, a strong enough push, we're gone. It's happening, right? You can't just will the dominoes to stop. No, all of the dominoes are going to fall once you press one. And if you guys have never seen people who do domino art with 250,000 dominoes, would highly suggest you can waste hours of your life just watching people who have wasted days of their lives setting up hundreds of thousands of dominoes. Anyway, you push one, they all go down, okay? So it's an all or nothing effect, and we're going to talk about that with the neuron shortly. So once it starts, it doesn't stop. It propagates, right? It goes through the entire row of dominoes, and then if you want to do it all over again, you have to meticulously go and set them all back up again. And why we do this, I will never know, okay? But they need to be all reset so that we can push them down again. Right? And this is going to be really, really similar. So if you can remember this basic principle, this is going to be really, really similar to how our neurons work. Okay, so the connection here then is that when we start, it starts by protein channels opening up. Okay, and once one protein channel starts opening up, it's going to be an all or nothing response. We're doing the darn thing. We're sending the signal. Okay. Then we propagate, so we have a positive wave, and I'm going to explain this in a second, but our neurons are actually negatively charged. Okay, so we have a positive wave rushing down our axon, again, just like the dominoes, all falling and hitting one another. And then we have to reset, so we do something called repolarize the membrane in order to be able to send another signal. And thankfully, our body does this very, very quickly, right? But we start, we propagate, and we reset just like if we were knocking over some dominoes, okay? Here's what it actually looks like. Okay, cool. So you need to be really, really familiar with what a neuron looks like and where we have concentration gradients. Okay, so what we can see here, so we have inside of the cell and outside of the cell. So inside of the cell, what you'll notice is there's lots and lots of little orange diamonds, and those are potassium ions. Okay, so our neurons are all going to be based on everything that we talk about with action potentials is all going to be based on potassium and sodium ions. Okay, so the inside of the cell is full of potassium, K+, and the outside of the cell is full of sodium, Na+. Okay, so positive charges outside, positive charges inside. Okay, but lots more sodium outside and lots more potassium inside. And the majority of the reason for that is because of this guy right here called the sodium-potassium pump. And when you think pump, you should think energy, like pumping iron or pumping your bike tires up, right? That requires energy. Okay, so the reason this requires energy, this is an example of active transport, and hopefully you feel really good about your like passive and active transport situations and concentration gradients, because that's all an action potential is. But with this active transporter, we're pumping 3 sodiums out where they don't want to be because it's already very crowded and full of sodium, and we're pumping 2 potassiums in where they don't want to be because it's already really crowded and full of potassium. So we're forcing things against their concentration gradient with our sodium-potassium pump. And it's one of our major users of our ATP supplies because obviously we need to put some energy in for this. You'll notice that there are some protein channels. There's more potassium channels than sodium channels just kind of leaking around. But then you'll also notice here that we actually have an inherently negative charge inside of the cell. So we have a lot of anions, a lot of negatively charged molecules in our neurons. So that's the major reason for that negativity. We're also pumping out more sodium than we're pumping in potassium. So we're losing more positive charges than we're gaining. So there are a number of reasons why the inside of our neurons are super, super negative. You have to be very, very familiar with the fact that there's lots of potassium inside and lots of sodium outside. The way that I tell my students to remember this, and this is also a pretty common analogy, so you may have heard of it before, is that our neurons are salty bananas. And the reason why is because the inside of the neuron, that's the banana, okay? Bananas are full of potassium, and our neurons are full of potassium. And then it's a salty banana, so we're sprinkling salt on the outside, and the chemical formula for salt is NaCl, so no, sodium is not salt, but close enough, and it's a good enough analogy that you can remember that there's lots of sodium on the outside and lots of potassium on the inside. And if you can remember that and you're familiar with your concentration gradients, then learning about this action potential is going to be pretty easy. Okay. So I told you guys that the neuron is actually negatively charged. Specifically, it would read, and we had a little, someone somewhere had a little microelectrode that they put inside the cell and outside of the cell, and they determined that the difference between inside and outside was a whopping negative 70 millivolts. It's actually a pretty strong charge on those neurons. So they're very, very negative. Again, a lot of anions, and we're losing a lot of positive charges. And this is going to play a key role when we're trying to propagate a signal from dendrite to axon and then to the next cell, or yeah, whatever it may be. Okay. Okay. So here's what's actually happening, because I haven't really explained much yet besides what the membrane looks like at rest and that it has a pretty negative charge. So the details that we're going to go into then is the details of the actual action potential, which is the process of sending the signal from one end of the cell to the other end of the cell. Okay, so we're going to go through a few of these main points, and I have pictures to describe all of them. So the cell at rest, we've already determined, is at negative 70 millivolts. Okay, so what happens when the cell starts to get a signal, and I'll show you this, is that the stimulus is going to increase the positive charge in the cell because we're going to start opening up sodium gates. And I'm going to show you that, but sodium is going to flow into the cell, and that nice positive charge is going to make it very positive. Once we reach a threshold, okay, it's negative 55 millivolts, and I'll explain this all in a much slower, more visual way in just a second. It's that all or nothing response. We push down the domino. The floodgates are opening up, and we depolarize the membrane. So the membrane is very polar, very negative, right, when we start. So we depolarize, meaning we're getting less and less negative. We actually get up to about 40 millivolts, positive 40 millivolts. So we go up almost 110 millivolts by opening up those sodium channels. Then we're going to go through this process of the sodium channels closing and the potassium channels opening. And when the potassium channels open, because this is a salty banana, and I'll show you this in a second, potassium is going to flow out. Potassium is a little bit slow, okay? So it's actually going to keep those, or the potassium channels are a little slow. So we're going to keep those potassium channels open for just a little too long. Gets us slightly more negative than negative 70. We call that hyperpolarization, and I'll explain that in a little bit. and then we go back up to resting. So this is the process that we're looking at. This is literally how we think and we move and we do everything that we do, because our neurons are going from negative to positive back to negative and then sending a signal at the end, which is really, really crazy, but kind of interesting. I always love the nervous system because your neurons are teaching you about themselves, right? Your neurons are firing right now in order to learn how a neuron fires, which is kind of funny. I don't know. My students have to put up with me every day. It's a crime. Okay, so let's look at this in a little bit more detail then, because I'm sure you have some questions, because this is not super easy to understand. So sorry that these are a little blurry, but in our resting state, right, we have our sodium channels and our potassium channels. They're pretty much closed up. Maybe a few are getting through, but mainly the sodium-potassium pump is pumping sodium out and pumping potassium in. So we have lots of potassium inside, lots of sodium outside, and we have that negative 70 millivolt charge. Then something miraculous happens. And we start to depolarize the membrane. So here's what happens. A number of things could happen. Sodium channels start to pop open. And there are a couple things that could trigger sodium channels to pop open. One, and we'll talk about this kind of in a backwards way at the end of this, is a neurotransmitter. So neurotransmitters are chemicals. You've probably heard of them before: acetylcholine, dopamine, serotonin, epinephrine. So they're little chemical molecules that are going to signal things, okay? And neurotransmitters actually bind, we call them ligand-gated channels, to sodium channels at the end of a dendrite. And when they bind, sodium channels pop open, right? Life is good. Sodium is going to come rushing into the cell. Why? Because there's a heck of a lot more sodium outside of the cell than inside of the cell. And remember that all molecules like to move from a high concentration to a low concentration. And so therefore, they're going to zoom themselves into the inside of the cell, the banana part, not the salty part, because that is where there's a lower concentration of themselves. Now, when this is happening, we're adding lots of positive charge into the inside of the cell, which is why during depolarization, we start to get super, super positive, okay? Because all of these positive charges are coming into the cell where there's a lower concentration of sodium, okay? Depolarization, sodium channels open up, oftentimes because of a neurotransmitter. But that's just at one end, right? So we have this neurotransmitter, and so they pop open, wonderful. What about the rest of the cell, right? And so the rest of the cell is usually controlled by voltage-gated ion channels, right? And basically these guys, like when it's positive on the outside and negative on the inside as it is right now, they're closed off, right? Kind of like opposites attract. That positive end and that negative end stick together and they're closed. But once the cell starts to depolarize, once they sense positivity, they're going to open up, just like you open up when you're surrounded by positive people. You're like, let me share my life story with you. I'm ready to be a part of this conversation, as opposed to being more closed off when it's negative, right? So negative environment, which is at rest, those voltage-gated sodium ion channels, wow, are closed. But once it starts to get positive in there, right, and that's going to happen when those first channels pop open, they're going to pop open too. And now you can see the cascading effect, because once more sodium channels pop open, more sodium comes in, we get more positive, and the voltage-gated ion channels next to them will start to pop open, right? And so on and so forth, and this is our tipping domino effect, okay? Once one sodium channel pops open, the rest are going to start to pop open because of that influx of positive charge that's bringing our cell all the way up to positive 40 millivolts. Good so far? Okay, you're quiet out there for the nervous system, but you're just pros at this. Okay. So we have this rising phase of our action potential, right? So all these positive charges. Now you'll notice that another voltage-gated ion channel has opened up and we're getting positive inside. Potassium is still unable to leave. The potassium channels are still closed. Okay, and we get our really positive interior up to positive 40. But then eventually, and we talked about this, so I'll zoom back really quick. Eventually, all good things must come to an end. The positivity must leave. So sodium channels are eventually going to close. That's going to be step 4 here. Okay, and if you know these 6 steps, you're good to go. You're golden, right? So once you look at them long enough and know the starting concentration of a neuron, you can figure out an entire action potential. But once sodium channels close up, potassium channels will start to open, right? And I'll show you that now. Potassium is going to leave. Why? Because it's a salty banana. So it's going to leave the banana and go towards the salty side, right? There's way more potassium inside of the neuron than outside of the neuron. And therefore, moving from high to low, potassium is going to start flowing out. Now, the sodium channels have closed at this point. We can see that here. Okay, and so potassium is going to freely leave the cell. That is going to repolarize or re-negativize, which is not a word, but we'll roll with it, the inside of our membrane because we're subtracting positive charges. We're losing the positivity of the potassium, which is also positively charged, and it's flowing out of the cell. And so our cell is going to start to get more and more polarized, more and more negative as more potassium ions leave. Okay? All right. So as I said before, we actually undershoot, right? So we go below negative 70 millivolts, and that's because our potassium channels are very slow. Just as they were slow to open up on the beginning, they're also going to be slow to close, which is going to allow us to get more negative than negative 70, okay? But that's not a bad thing because that little undershoot right here is going to prevent us from actually sending a signal back the other direction. Okay, it basically closes off afterwards so that we can't send a signal in the opposite direction back towards the head where it came from, right? Which I always think of as being like, hey, so-and-so, I have something to tell you. Oh, wait, never mind. And you're like, what? It's like the worst thing that a person could ever do to you when they tell you they have to tell you something and then say that you can't know it. That's basically what this would be. It's like, we're going to send you a signal. Oh, wait, never mind. We're going back to home base. Right? So in order to prevent that and just send the signal, right, we undershoot a little bit. We call that hyperpolarized because not only are we polar, we're hyperpolar, right? And then we're going to eventually reset the membrane potential using the sodium-potassium pump in order to get back to negative 70. Good so far? Cool. That's a really important slide. So, which one? All right, so what this kind of looks like from like a grander scheme, right? We have the action potential traveling down this axon here. So sodium comes in and we get nice and positive, okay? And then once sodium comes in, potassium is eventually going to leave, okay? But while this is happening, right, charge down the line because our voltage-gated ion channels are popping open, okay? And then that reset by potassium leaving is kind of following us. And then you can see the membrane will officially be reset by our sodium potassium pump. But this just shows kind of how that positive charge propagates down the membrane and then how the negative charge follows right behind because of the potassium channels opening up afterwards. And then we have our official reset. And the reason we need official reset, the reason we need that sodium potassium pump so desperately is because what we've done here is put a high concentration of sodium on the inside of the membrane and a high concentration of potassium on the outside of the membrane. And while we still have our negativity inside of our cell, we've reset in that capacity. In order to have our next action potential, we are going to have to put the salt back on the outside and the banana back in the inside, the sodium out and the potassium in. And remember, that's what our sodium potassium pump does, is pump 3 sodiums out and 2 potassium in so that we can reset our concentration gradients. Questions about that? Okay. So I already mentioned this, the final reset, we're using ATP, we're pumping 3 sodium out, we're pumping 2 potassium. Easy. Okay. Almost done then, because you guys were so quick. Although I can put up a few key neurotransmitters as a last slide here. But so at the very end, right, so we've sent the signal all the way through, and now we've made it to the end of the axon terminals. And you're like, what does this positive charge rushing through the axon have anything to do with us thinking or moving? The answer is all lies in these little synaptic vesicles all the way at the end. So as our action potential, which you can see by these arrows, approaches the end of our axon, we're actually going to go through a process where these synaptic vesicles, so vesicle, I always think vesicle, vehicle, so these little cellular vehicles that have neurotransmitters on them are going to make it all the way to the end. They're going to be stimulated by the action potential to release their contents into this area, which we call the synaptic cleft. And that's going to be the gap between the end of an axon and either its target cell or a dendrite of another axon. Okay, so there's this little gap. It's kind of like I was telling my students, like Red Rover, right? You got to run to the other side. And a lot of them had never played Red Rover before, so maybe you have. But these neurotransmitters are going to be released. They're going to kind of scurry across this gap, and then you can see we have these receptor proteins on the outside, right? Here's a closed sodium channel, and once this neurotransmitter binds to it, it opens up that sodium channel, and we're going to see a reaction occur then, right? That's the very, very starting aspect of how an action potential would be propagated. In this case, the neurotransmitter is acetylcholine because our target is a muscle cell, and acetylcholine is always going to be the neurotransmitter that has our muscles contracting. Okay, so when they get this action potential, when the acetylcholine travels across the synaptic cleft, binds to the other side, it's going to cue these muscle fibers to contract. The cells that you'll see will shrink up, and you'll have the muscle, and you'll be moving or talking or breathing, or your heart will be pumping, or any number of things as a reaction of this action potential through this neuron and then these neurotransmitters at the end. Cool? Okay. Let me put out just a few more important neurotransmitters that you might want to know about. This is actually a good little graphic here that I can post for you. The link in here, as opposed to putting it on the slide, which will take a second. Okay, you can take a look at adrenaline, also known as epinephrine, and noradrenaline, also known as norepinephrine. So we have adrenaline, which is our fight or flight. Epinephrine is our fight or flight. So stressful or exciting situations, it's going to increase our body, get us amped up. It's also what can put your body into shock, right? Or you could get like a broken arm because you're running away from a mountain lion perhaps, and you don't even feel it because adrenaline is rushing through your body. And so that's actually a really important evolutionary, a lot of these neurotransmitters are evolutionary masterpieces, right? Because not feeling pain in the moment so that you can get yourself out of the situation is really, really important. Norepinephrine is going to help more with concentration. Dopamine is like our pleasure sense. It's going to be like addiction, movement, motivation, like happiness, joy. Dopamine is that. Serotonin is going to be more involved with our sleep cycle. So if you've heard of melatonin before, it's just kind of like a artificially made serotonin situation. GABA calms us down. Acetylcholine is going to be for muscle and also thought and learning. We have glutamate for memory. You can take glutamate supplements, actually. People do take glutamate supplements if they're like relatively forgetful. So if you feel like you're forgetful, you can just pound some glutamate. And then all sorts of endorphins. It's going to jazz our body up. So all of these signals, you can see also the structure of them, which is pretty interesting. They get passed through the synapse and stimulate sensory reaction on the other end. So we want to have any questions before we end tonight? Nothing? Oh my goodness. Okay. Well, those are the major parts of the nervous system. Hopefully this was helpful and you understand a little bit more about action potentials, the different cells of the nervous system, etc. Next week, I believe, we're talking about homeostasis. Oh, example neurotransmitters. Yeah, so if you pull up in that slide, it has all the information that I just went, or that link that I just sent. It has all that information in it. But I can certainly go through it in more detail. So adrenaline is going to be, and if you click on the picture, it makes it bigger so that you can take a look at it. So adrenaline, also known as epinephrine, is going to be fight or flight. So adrenaline is really, or epinephrine is really going to amp up our responses. So increased blood flow, increased heart rate. So increasing blood flow increases oxygen. So increasing oxygen increases our capability to go through cellular respiration. So all these things would be really, really important when you're really excited or really stressed or running away from a mountain lion or trying to make the game-winning shot or something like that. That feeling of your heart pounding and your palms sweating, that's epinephrine running through your body and making everything, increasing that heart rate, increasing your breathing so that you're getting more oxygen, everything's activated and you're ready to rock and roll. Norepinephrine is going to be involved also in the fight or flight response, but that it's going to have essentially the opposite effect. So we're going to contract our blood vessels. I guess we increase heart rate, but it's also going to be involved in your attention span. Okay, so those two are going to be involved in that major process. So dopamine is what makes you happy. So people can take artificial dopamine. It's an illegal drug, but it's what makes you happy. And it's also what makes you addicted. So that's why dopamine is incredibly dangerous, because not only are you taking something, but you're taking something that also leads to addiction. But it also increases your motivation. So when you're really motivated, when you're feeling really positive, that's going to be dopamine that's firing in your brain. Serotonin is kind of the opposite of that. It calms you down. It's going to contribute to your kind of happiness, but also helps with your sleep cycle. And the really interesting one about this is it's affected by exercise and light. So serotonin levels can be low when you're not exercising and when there's no sun out. So in the wintertime, if you take a little hiatus, that's going to be contributing to you feeling less happy. So seasonal affective disorder is going to be oftentimes because of a decrease in serotonin. But if you continue to exercise and try to get out in the sun still, if you can, that's going to help you out. GABA helps calm us down. It's kind of like the opposite of adrenaline. It's going to just kind of chill us out, and it decreases the firing of our central nervous system. And if you don't have a lot of GABA, then it can lead to anxiety. So if you look at anti-anxiety medications, oftentimes they'll mention GABA. Acetylcholine we've talked about a lot, but that's going to help mainly with muscle or with firing off muscles. But it's also involved in thoughts, learning, and memory. So a lot of these have dual purposes. The different responses come from the different cell that they're targeting. So targeting a brain cell is going to be different than targeting a muscle cell, and you're going to have a different response there. Glutamate helps with memory. And as I said, you can take glutamate supplements. And then endorphins are what keeps us happy. They're released during exercise. They make you excited. All those good things. Similar to serotonin and dopamine, they're going to help pep you up. Cool. Hopefully that helps. Awesome. Well, that is all I have for tonight. Tune in next week for homeostasis. We'll talk about how our body maintains all of our body temperature, blood sugar, everything else, water levels, everything else in that our body does without us even knowing. We'll talk about positive and negative feedback loops, all sorts of good things. So definitely tune in. Sign up for a 5able Plus membership so you can watch all of the videos that I've made since September. Lots of good information there. And other than that, I hope you have a lovely week. I look forward to seeing you next week. And tune in. Bring your friends because it's free. Might as well. All right, y'all. See you next week.